Okay, in this video we're going to be talking about the cell. So let's just show you what some of these things look like first, and then we'll get into what some of the functions are. So this is a cell model that I use to teach the different organelles. So all the way here on the outside is the cell membrane. Sometimes they call it the plasma membrane, which remember is a phospholipid bilayer. It is a phospholipid bilayer. All the way on the inside is the nucleus. That's the clear translucent layer. The orange pit that we see in the center, that's the nucleolus. So you have a nucleolus on the inside. Superficial to it is the nucleus. Then we have a nuclear envelope, which is the white structure surrounding it, with these nuclear pores. Then we have an endoplasmic reticulum, which is this aqua color that is continuous with that nuclear envelope, with these white speckles of salt on it, right? These white speckles are called ribosomes. When we have ribosomes that are fixed to the endoplasmic reticulum, it has a rough appearance, so it's called a rough endoplasmic reticulum. Where we don't see the ribosomes fixed to the endoplasmic reticulum, it's called a smooth ER, or the smooth endoplasmic reticulum. Since the structures are different and they look different, they function different. The smooth endoplasmic reticulum and the rough endoplasmic reticulum are very different in function. Okay, the cytosol is the clear fluid between the organelles. The fluid with the organelles is called the cytoplasm. The cytoplasm. Let's go back here. These are the centrioles. These spindles here are centrioles. The centrosome is this black circle here. These are involved with cellular division. When you look at mitosis, you'll learn about these spindles and the centrioles. Here is the Golgi complex, also known as the Golgi Apparatus. Here is the mitochondria. Then we have lysosomes and peroxisomes. Lysosomes and peroxisomes. Again, they may look similar, but their structure is different, which means they function different. Okay, those are the main organelles. There are some others that this model doesn't show, like the cytoskeleton of the intermediate filaments and the microtubules and the cilia and villi. I'll show you those on a different picture. So remember, the organelles, they just sound like different, like small organs. These are specialized structures within a cell, and they have certain type of shapes and structure, which will then dictate its function. Some are involved with cell growth, some with cell maintenance, and others with reproduction. The centrioles are these paired organelles that are composed of microtubules. Microtubules are a large type of protein, and they form the spindle apparatus, which is involved during cell division, known as mitosis. I showed you the centrosome, which was that black dense body. The cytoskeleton is a network of protein elements like microtubules and microfilaments and intermediate filaments that are involved in providing structural support for the cell. The plasma membrane is that outer part of the cell membrane. It is a phospholipid bilayer. It controls what goes in and out of the cell. Extracellular fluid is fluid outside of the cell, whereas the cytoplasm contains the cell organelles and the fluid. It's everything from the cell membrane to the nucleus. Everything that you see within that level is going to be the cytoplasm. The cytosol is the intracellular fluid, whereas the fluid outside of the cell is extracellular fluid. Microvilli and cilia look similar, except the microvilli are much, much smaller. They're these finger-like extensions. They're really involved with increasing the absorption and increasing surface area, whereas the cilia, these are involved in helping to move around. They whip around, so they help to move the cell around, and they can also move debris around. We will see lots of cilia located in the trachea of the respiratory tract. Ribosomes are complexes of RNA, which stands for ribose nucleic acid. So they're complexes of RNA and protein that are found in all cells. The function of the ribosomes is to produce protein, synthesis of proteins. There are two types of ribosomes, fixed ribosomes and free ribosomes. The free ribosomes are roaming around freely in the cytoplasm, and they're going to manufacture protein for the cell, whereas the fixed ribosomes are attached to the endoplasmic reticulum. That's where it gives it that rough appearance called the rough endoplasmic reticulum. The free ribosomes produce protein for the cell. The rough endoplasmic reticulum is going to produce protein to be shipped out of the cell, or at least can be used to regenerate any damage to protein within the cell membrane. The nucleus is the control center of the cell. It is the location for your genes. It contains the DNA. The DNA, this is the template or the information, right, for what proteins need to be produced. It doesn't produce the proteins, it just has the instructions for what proteins need to be produced. The nucleolus is the site of the ribosomal RNA synthesis. Okay, and remember the nucleolus is deep to the nucleus. Now let's look at the difference between the smooth and the rough endoplasmic reticulum. The rough endoplasmic reticulum was continuous with that nuclear envelope. Remember, you had a nucleus, you had the nucleolus deep to it. So you had the pit, which was the nucleolus, then you had the nucleus, then you had nuclear envelope, and the rough endoplasmic reticulum was continuous with that. And it's involved in the synthesis of proteins, glycoproteins, and phospholipids. The smooth endoplasmic reticulum has no ribosomes at all. Okay, it doesn't synthesize proteins, but it's involved in carbohydrate metabolism and synthesis, as well as modifying lipids or fats. That's the smooth ER. The Golgi apparatus consists of a bunch of those flattened saccules, and they're involved in sorting and modifying of proteins. So the Golgi apparatus can make, the rough endoplasmic reticulum can make certain proteins, and then they ship it off to the Golgi apparatus where they modify them. And they package them and then export them out. Okay, and some of those proteins that are packaged can be shipped outside of the cell. Maybe it's a digestive enzyme, maybe it's a neurotransmitter, maybe it's a hormone that needs to be shipped out, and then it uses a method called exocytosis. Sometimes it produces a protein that needs to be utilized to regenerate and repair damaged proteins in the cell wall or in the cell membrane. Okay, and sometimes the Golgi apparatus creates lysosomes. And lysosomes, just below it, is a vesicle that contains digestive enzymes, and they are responsible for helping to rid and clear out any cellular debris. The lysosomes contain not just digestive enzymes, but lots of hydrogen ions. And when hydrogens are dumped onto something, and you increase the amount of hydrogen, you start to decrease the pH. You make it very acidic. And when the pH drops below 5, it just destroys whatever it is the hydrogen was being dumped on. So lysosomes are really important for clearing out debris. Peroxisomes, these are vesicles that are designed to detoxify harmful substances or toxic substances. So peroxisomes are found throughout the kidneys and throughout the liver because those are the organs that detoxify, especially alcohol and drugs, anything harmful to the body. Peroxisomes are utilized to detoxify those, and free radicals, also known as reactive oxygen species, which are abbreviated ROS, reactive oxygen species. They help to neutralize dangerous free radicals. When you have oxidants or free radicals roaming around, you could damage your DNA. And that's where oxidative damage could be so damaging, but that's why we eat foods with antioxidants to kind of neutralize those. Peroxisomes are also involved in something called beta oxidation. Beta oxidation is the term used to describe the breakdown of fatty acids. Okay, the beta oxidation is that every time it breaks down the bonds, every 2 carbons, at every second carbon, right? The first is alpha, the second is beta. That's beta oxidation. The mitochondria is the site of adenosine triphosphate production or energy production, but the energy fuel source is called ATP, adenosine triphosphate. In order to produce energy, the body needs glucose, and it also needs oxygen for that. So some of the questions that I've seen pop up, pretty straightforward. Looks like we do have some sentences here, and what's in blue is missing, so you'd have to drag what you see above down. So the blank modifies, packages, and sorts newly synthesized proteins. Well, we know what synthesizes the proteins is the endoplasmic reticulum, but what modifies them and packages them is the Golgi apparatus. So that's here. The green here is the Golgi apparatus. The rough endoplasmic reticulum synthesizes proteins for secretion out of the cell and for utilization in the cell. That's the rough ER. The mitochondria synthesizes ATP, adenosine triphosphate, for energy of the cell. And lysosomes digest macromolecules, microorganisms, and all damaged organelles. Remember, lysosomes can be produced by the Golgi apparatus. They have these digestive enzymes, and they dump hydrogen on whatever it's trying to destroy. Okay, now here it looks like we have to drag these items next to the appropriate box. If it's involved with the cell surface, if it's involved with the nucleus, which is here, or inside the cell for the cytoplasm. So this is basically testing your understanding of the structure. So if we're talking about nucleolus, DNA, and chromosomes, those are all linked to the nucleus. If we're talking about the cytoskeleton, those protein filaments, the intermediate filaments, the microtubules, that cytoskeleton is part of the cytoplasm. Mitochondria and Golgi apparatus, those are just organelles here in the cytoplasm. And then along the cell surface, like here, these are microvilli. Here, these are cilia. So cilium, microvilli, and the plasma membrane, those are all part of the surface of the cell. So pretty straightforward, right? Nothing to really memorize here, just being familiar with the information. Now we're looking at the phospholipid bilayer. So this is extracellular. This is outside the cell. This is inside of the cell. This is one phospholipid layer, and here's the other phospholipid layer. The phosphates are these heads or the spheres. Those are the phosphate portion, and the lipids or the fats poke inside. So here's a phospholipid layer, here's a phospholipid layer, so we say it's a phospholipid bilayer. The phosphate portion, which faces the extracellular fluid, and the phosphate portion here, which faces the intracellular fluid, we say it's hydrophilic because it likes water. Inside the membrane, right here, would be hydrophobic. Okay. Some of these lipids are straight, they're saturated, and some have a kink or a bend to it, meaning it's an unsaturated fat. You need both saturated and unsaturated fat in your diet. Why? Because the cell membranes are made of saturated and unsaturated fat. It's also made of carbohydrate. It's also made of protein. So you need fat, carbs, and protein in your diet to help make up a healthy cell membrane. So here is an integral membrane protein. Integral means it kind of goes through the full membrane from one side to the other side. If it doesn't go through and just hangs on the outside, like here, that's a peripheral membrane protein. Here are glycoproteins. These glycoproteins are made up of protein and carbohydrate together. Glycoproteins are important. Some of them act as like cell identity markers, that type of thing. Like a flag that says, hey, you know, I'm a kidney cell, or I'm a muscle cell, or I'm a liver cell. It identifies itself. Here's a membrane protein. Here's a channel, a membrane channel pore. So when this opens, maybe calcium. If this is a calcium channel, calcium goes through here. If this is a sodium channel, maybe sodium goes through here. So proteins, some of these channels are actually made up of proteins. So now let's look at what we would have to drag. We see that we have carbohydrates in the membrane. We have lipids, which are fats in the membrane. We have proteins. So you have protein, fat, and carbs. So now we have to drag and drop what is best associated. So let's look at proteins first. So proteins can act as channels. They're membrane channels. Proteins can also act as enzymes. Enzymes are proteins. And proteins can actually be receptors as well. So many receptors on organs are made up of proteins. When we look at fat, membrane lipids, we have cholesterol. About 15 to 20% of the cell is cholesterol. We do need cholesterol in our diet. Our liver is the main organ that makes cholesterol. It primarily makes it at night. And this is why when people have high amounts of cholesterol, they're supposed to take these statins at night because that's when the liver makes it, like simvastatin, Crestor, Lipitor. Those are all anti-lipidemic medications. But you have to be careful with them because the brain needs cholesterol and cell membranes need cholesterol. So one of the black box warnings on these anti-lipidemic meds are dementia-like symptoms because the brain does need cholesterol. We mentioned that the phospholipid layer has a hydrophilic head and a hydrophobic tail. Remember, the hydrophilic was the sphere and the hydrophobic was the saturated and unsaturated tails. And the lipids compose most of the plasma membrane, for sure. Then you have the carbohydrates. The carbohydrates are involved in immunity to infection. It's involved with the glycocalyx. The glycocalyx, if you've ever tried to catch a fish and you feel the scales of the fish, how slippery it is, that's what the glycocalyx is. Right? It doesn't allow things to stick to it too much. It allows things to pass by because it is so slippery. Membrane carbohydrates are involved with transplant compatibility as well. Okay. Here, the green region, these are going to be cilia. The large ones are cilia. These are microvilli. Microvilli are designed to increase the surface area of the cell, and it's also used for absorption. Here, the highlighted portion, in this case, is the mitochondria. the mitochondria is involved in producing adenosine triphosphate, ATP. So let's look at energy production. So it says, put the sentences in order to describe the process of glycolysis. Whenever we see the word lysis at the end, it means to break down. So glycolysis or glycolysis is saying, how do we break down sugar? Okay, so the oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. So glucose is like a six-sided sugar. And when you take that six-sided sugar and you split it in half, you get these 2 and 6 carbon sugar, I should say. So when we divide the 6 carbon sugar in half, you're going to get 2 3 carbon sugars. You got a carbon here, a carbon here, and a carbon here. Then you get a carbon, carbon, carbon. So when they're together, you have 6 carbons. When they're separate, 2 3 carbon sugars. These 2 3 carbon sugars are called pyruvate. And as long as oxygen is available, pyruvate can enter the mitochondria and be broken down further. If oxygen isn't available, pyruvate becomes lactic acid. Okay, and that's what gives people that burning sensation, kind of. It's one of the things when you're running out of oxygen and then you're training and exercising, you get a burn, you kind of drop weights, you breathe, you get oxygen levels up, and then the burn goes away, and then glucose has been regenerated and you're ready to exercise again. So the initial steps in glycolysis are the addition of 2 phosphates to the glucose. I think they call it glucose 6-phosphate. So it just attaches a phosphate to it at first. The result is a 6-carbon sugar, fructose, 1,6-biphosphate molecule, and 2 low-energy ADP molecules. ADP is adenosine diphosphate. When you see AMP, it's adenosine monophosphate. So you have AMP, adenosine monophosphate. You attach a phosphate to it, adenosine diphosphate. You attach a third phosphate, you have ATP, adenosine triphosphate. Okay, the 6-carbon sugar diphosphate molecules then split into 2 3-carbon molecules, and each of the 3 carbon molecules is converted through a series of steps to pyruvate or pyruvic acid. Under aerobic conditions, the pyruvate is further oxidized to produce more ATP. That's under aerobic condition, meaning in the presence of oxygen. But if you're out of breath and there's anaerobic conditions, the pyruvic acid is converted into lactic acid. Okay? So this ability to convert pyruvic acid into lactic acid and lactic acid into pyruvic acid is really important. The body has the ability of going back and forth depending on whether or not oxygen is readily available. That's when we tell people, oh, they're exhausted, you know, go sit down, rest. They start to breathe, they get their oxygen levels up, and then lactic acid is converted back to pyruvic acid. Okay? And then here in the center, we have the nucleolus, then you have the nucleus, you have that nuclear envelope with the nuclear pores, and then you have the rough endoplasmic reticulum that's there. And what's the function of the rough endoplasmic reticulum? It can synthesize proteins and it can use them to kind of export them for the cell or outside of the cell. And then here is the Golgi apparatus or the Golgi complex. What is the Golgi complex for? It's going to modify and package and export the proteins that the rough endoplasmic reticulum produced. Okay, so the rough ER produces it, and then it ships it to the Golgi complex or the Golgi apparatus where it modifies those proteins. And then you could see these vessels here Maybe one is a lysosome. Maybe another one is another vesicle that's going to be open up and release its proteins to outside of the cell through a method called exocytosis if it's a neurotransmitter or some sort of hormone or something, okay? This is the mitochondria. This is the Golgi apparatus. Here's the rough endoplasmic reticulum, and then there's the smooth ER. Remember, the smooth ER is for carbohydrate metabolism and synthesis, as well as lipid metabolism. Here are the microvilli. Here's the cilia. Now we start to see some of the cytoskeletal structures. So the microtubules are the larger proteins. Then we have the microfilaments are the thinnest proteins. And then you have the intermediate filaments, which are, you know, bigger than the microfilaments, but smaller than the microtubules. Here are the centrosomes, composed of those centrioles, which are involved in cellular division. And then here's the plasma membrane, that phospholipid bilayer. Here's the nucleolus. Here's the nucleus. These are the nuclear pores, which is where the ribosomes and that ribosomal RNA is going to squeeze right through there. These are free ribosomes. When those free ribosomes are attached to the ER, it's a rough endoplasmic reticulum. Okay. Okay, this one, I think we did already, right? I think we did this one. This one popped up a second time. This was knowing whether certain structures are part of the cell surface, the cytoplasm, or the nucleus. We said if it's DNA, chromosomes, or nucleolus, those are all associated with the nucleus. If it was the cytoskeleton, like the microfilaments, the intermediate filaments, or microtubules, the mitochondria or the Golgi apparatus or any other organelle for that matter, that's part of the cytoplasm. And then the cell surface would be the plasma membrane, the microvilli, and the cilia. Okay, this structure here, this from here to here is the nucleus. This is the heterochromatin. These are vesicles. The darker structures are peroxisomes for detoxification. These are the mitochondria. These are, the clear ones are vesicles, right? We don't know what type of protein may be in here or what its use is, but these are the peroxisomes for, you know, beta oxidation, for detoxification of reactive oxygen species and detox. So we'll see them in high amounts in the liver. And that's why when people drink a lot of alcohol or abuse drugs, they start to burn out these peroxisomes, and then the liver becomes damaged. Someone can get cirrhosis of the liver. Okay.